Good morning, good evening, ladies and gentlemen. Welcome to 40th APAMS, the 40th Anniversary Annual Scientific Meeting and Symposium of PAASE, or the Philippine-American Academy of Science and Engineering. I'm Gisela Concepcion, 2020 PAASE President and the host of this opening plenary session. As we commemorate the 40th foundation year of PAASE in the period of the global COVID-19 pandemic, we are confronted with serious challenges and unique opportunities. With the rest of the scientific community, we are called upon to contribute to understanding the evolving situation of COVID-19 from the scientific standpoint as basis for finding ways to stem its devastating impacts. And therefore, we aptly begin this conference with an opening plenary session today on the subject of COVID-19. Thank you for joining us. Stay with us through the rest of the month-long conference with the theme, Outreach to Philippine Regions. 40th APAMS is unprecedented. It runs for 4 weeks every day, Mondays to Fridays, starting at 8 a.m. Philippine time, from today, July 20 until August 14. It features 6 plenary sessions, 30 parallel sessions, 4 poster sessions totaling more than 200 presentations, and 8 group discussions on science advocacies. As you will note in the scientific program, 40th APAMS, okay, you see us now. 40th APAMS showcases the science-based endeavors pursued not only by PAASE members, but also by champions and practitioners of science from other sectors in urban and rural areas of the country who could become PAASE's partners. 40th APAMS is unique. The host of each parallel session planned his own session and invited special speakers in his area of research. Each parallel session is as important as each plenary session. Our group discussion leaders are formulating key discussion points and a framework to share with participants during the discussions. Okay, can I ask everyone to mute their microphones, please? With a rich repertoire of presentations and discussions, 40th APAMS provides the unique opportunity to bring us together to jumpstart the conversations on science-based problem solving and productivity in the country. We note that a tagline of PAASE is Agham at Kaalaman para sa Bayan. Registration in 40th APAMS is free, and you need only to click the Zoom link of each plenary or parallel session or group discussion to register. One thing I think each of us has learned from COVID-19 is the value of parsimony. The virus, so intelligent, so efficient, possesses so little, yet does so much. We ourselves now learn to retain mostly the barest essentials and shed the superfluous peripherals. We are maximizing our interactions on air, liberated from time and space, able to focus on the substance of our messages, able to communicate them clearly with the aid of pre-recordings and with slides that can be easily read from our desktops. We are becoming more efficient at little or no cost, except for the cost of our commitment, our talent, and effort. So let us make the most of 40th APAMS. We are deeply grateful to many PAASE members and friends who are making the entire month-long celebration possible. In closing, let me say, this is the best of times, in the worst of times, to celebrate 40 years of science and engineering practiced by generations of PAASE members. We thank PAASE's founders and honor them by keeping PAASE growing and vibrant. In particular, We would like to thank all our speakers who will share their knowledge with us in this conference. We hope to shine a light for science to lead us out of the darkness of the current crisis. Let us hope that in this long, arduous struggle, we can celebrate our victories and endure and learn from our failures. Thank you. Let me now turn over control to Dr. Serafica, who will lead the panel discussion of PAASE members who are newly elected to the National Academy of Science and Technology, or NAST, on the subject of COVID-19. Al?
Thank you, Giselle. Thanks. That was a very nice welcome. Hello, fellow PAASE members. Hope everyone's doing well and healthy. And we're looking forward to seeing you in person soon. But for now, we'll have to stick with this format. Today, we've asked 5 of our members, including me, to briefly just describe what we've been doing related to COVID-19 in this time of lockdown in the Philippines and in the United States. But I think we only have one representative from North Carolina, that's Francis. So that the intent was really just to describe what one person can do to help in our effort to fight the virus and the pandemic. And I think after each of one has a time to kind of present and describe what they've been doing, I ask everyone also to come up with a wish list in terms of what we would want to do moving forward. So I think with that introduction, we're gonna have Ann Villalobos first, ladies first, to go ahead and describe her efforts. Then it's gonna be Windell, and then Francis, Chris, and then me. So take it away, Ann.
Good morning or good evening. There are 2 basic ways to develop vaccines against COVID. The first one is by the traditional method, that's the live attenuated virus, or the inactivated virus. And then there's the second one, which is more advanced and novel. That's based on biotechnology, where you have the nucleic acid based, either DNA based or RNA based. And then the viral vector vaccines, wherein you have replicating or non-replicating expression vector, and then virus-like particle, and then subunit of SARS-CoV-2. Now for this, there's a total of 185 vaccine candidates that's being studied right now. And for therapeutics, it could be the antibodies, like the cocktail of neutralizing antibodies that Regeneron is developing, and obviously the antivirals like remdesivir, cell-based devices, or repurposed drugs like dexamethasone and hydroxychloroquine. Next slide, please. Next slide. Next slide. Okay. Now for vaccines, it is so impressive that the world has joined together to develop vaccines against COVID. And with the cooperation from private, government, NGOs, vaccines have been developed. And this time, it's something unique because it's developed by the new scale and new speed. New scale in a way that millions of doses are manufactured per day in order to meet the clinical needs. And the hurdles there are, you have to manufacture also millions of vials and syringes, and where the contract companies take place also. That's the contract bulk manufacturing that's needed. New speed in a way that for the uniqueness of COVID, that means that the vaccine development has to speed up. That means that the sponsors who develop the vaccines review their data, interpret them within themselves, that's internally, and then consult with regulatory bodies, and they have to make the decisions forward at their own risk. So because of that, the COVID-19 vaccine development is only for 12 from molecule to market. Now, when you compare that with the industry normal development, that's 10 to 18 years. So that's very impressive right now. So there's a lot of opportunity for the sponsors. Next slide, please. Now with this, I have here the front runners for vaccines and therapeutics against SARS-CoV-2 or against COVID-19. And by the end of the year and early next year, approximately 3 to 4 vaccines will be available either by emergency use authorization or in the market of some cases. But the question is, will it become normal when vaccines are available? Now we have to address this, the problem with mass allocation and distribution. What's the capacity and the manufacture of these vaccines, and can they supply the whole world at once? And that means that there are vaccinated and non-vaccinated population, which is also dangerous. And the other concern is, do these vaccines have long-lasting immunity or just a year like the flu vaccine? And will they be effective in older populations? Because usually it's effective right away for younger population, but the older population may not be that effective. So that means that the new normal that we will have, even with the vaccines, we still have to have strict compliance in terms of still wearing our face mask, social distancing, and hygiene. Thank you.
Thank you very much, Anne. That was really a very nice and concise description on where we are on vaccines and development. Clearly, we're compressing time and space right now with our efforts in the scientific community to come up with a solution towards our common enemy. And those questions really are very relevant and difficult to answer at this point. And I think we'll just have to cross the bridge when we get there. For the next speaker, it would be Windell from UP, and he will be describing his efforts in the detection and diagnosis or his for COVID-19. Windell?
Yeah, thank you very much, Al. Good morning, everyone. I'll be discussing about salivary diagnostics for COVID-19. Take note that this is a prospective study. Okay, as we all know today, rapid and accurate diagnosis of COVID-19 is crucial in controlling the outbreak in the community and, of course, in the hospitals. Currently, nasopharyngeal and oropharyngeal swabs are the recommended specimen types for the RT-PCR detection of SARS-CoV-2. The collection of these specimen types, of course, would require close contact between the healthcare workers and patients and would pose a risk of transmission of the virus. It would also cause discomfort and may also cause bleeding, especially if the patient is suffering from thrombocytopenia. Now, these collection systems can also induce coughing and sneezing, generating aerosol, which can obviously transmit the virus. Hence, nasopharyngeal and oropharyngeal swabs may not be desirable for sequential monitoring of viral load. On the other hand, of course, we know that blood is the specimen of choice for antibody detection. Well, we also know that COVID-19 is associated with human-to-human transmission, and SARS-CoV-2 was recently detected in the saliva of infected patients. Saliva does have a significant role in the human-to-human transmission. Hence, salivary diagnostics may provide an easy and cost-effective point-of-care platform for quick and early diagnosis of COVID-19. Saliva is secreted from the human salivary glands, which contains minerals. You have their electrolytes, buffers, enzymes, enzyme inhibitors. You have growth factors, cytokines, also immunoglobulins. In particular, you can see their secretory IgA, mucins, and also glycoproteins. Now, interestingly, the saliva that is harvested from the space between the teeth and the gums is enriched with gingival crevicular fluid, or GCF. Now, the composition of GCF resembles that of serum and is enriched with antibodies. So you have there your IgG, IgM, and also your IgA. Okay, saliva has been studied thoroughly as a potential diagnostic tool, and it is expected to become a substitute for other biological fluids, such as serum, in disease diagnosis. So saliva can be used for the direct detection of the SARS-CoV-2 virus and the quantification of the specific immunoglobulins produced by it. Now, saliva specimens, as you know, can easily be obtained easily, as the patient can actually be asked to spit into a sterile container. But of course, if you would like to do this in our own setting here in the Philippines, we have to standardize the sample collection. Now, the collection of saliva is non-invasive and greatly minimizes the exposure of healthcare workers to COVID-19. Now, according to studies, saliva has a high consistency rate of greater than 90%, with nasopharyngeal specimens in the detection of respiratory viruses, including coronaviruses. Now, what are some of the advantages of using saliva for diagnosis? Well, of course, it is safer to administer than serum sampling because there's no need for use of needles. There's also no need for trained medical staff. Multiple samples can actually be obtained easily. Collection and screening can also be done at home. It minimizes the risk of cross-contamination. It requires lesser manipulation during diagnostic procedures compared to serum. And saliva does not clot and can be manipulated easily than blood. Next slide, please. Our next slide.
Which one? Please move that there.
Okay, so SARS-CoV-2 can be detected in saliva, as I mentioned, at high titers. So salivary diagnostics is a dynamic field that is being incorporated as part of disease diagnosis, clinical monitoring of systemic health, and to make significant clinical decisions for patient care. Now, I mentioned earlier about GCF and the presence of IgM, IgG, and IgA in serum against components of the virus, such as nucleoprotein. And you also have the action of antibodies against spike protein, in particular the receptor binding domain. It has been described in COVID-19, especially for those patients about 10 days or later after symptom onset, and they were able to clearly see this from the serum. Now, presence of these immunoglobulins detected in saliva correlated well with antibodies in serum, showing high sensitivity and also specificity. So salivary antibodies to source SARS-CoV-2 could serve as a non-invasive alternative to serological testing for widespread monitoring of SARS-CoV-2 infection throughout the population. So if saliva can support measurements of both the disease, the presence of the SARS-CoV-2 RNA virus, as well as antibodies against the SARS-CoV-2, this sample type could provide an important opportunity to monitor individual and population level SARS-CoV-2 transmission, infection, and immunity dynamics over place and time. Thank you.
Thank you very much, Wendell. I've been very excited about actually the saliva detection capability. I've heard of an emergency use authorization in the FDA as early as April 15. In Rutgers University, a professor there has been doing some tests, and we contacted him. He was doing about 10,000 tests a day, and I can't wait to have access to that data to guide us where we need to go from here. I think I remember Dr. Asuncion as well mentioned a group here in the Philippines. Of course, for our fellow members, Wendell has been able to develop a LAMP saliva-based detection for dengue, and that's where his interest of extending that capability to COVID-19 has emanated from. So I think we're going to move on from the detection and the vaccines to environmental monitoring. So we would like to, Richmond, if you can load the Francis de los Reyes slide, please. And Francis, I think you're all set to move forward. Once your slide is up, Richmond.
Okay, well, good morning. Thank you, Richmond. Thank you, Al, for the introduction. So I have been working in water and wastewater as an environmental engineer and microbiologist. And as early as March, yeah, actually, Richmond, if you can stay on presentation mode. As early as March, we were talking about how we can possibly detect SARS-CoV-2 in the environment. And in April, we were able to submit and get funding from the National Science Foundation in the US on SARS-CoV-2 in water. So this first slide actually summarizes the current knowledge that we have in terms of SARS-CoV-2. So the first point is that the virus is easily inactivated in the environment, in water, and on surfaces. And that's why it's easy to kill the virus. It's an enveloped virus that is actually easier to inactivate with surfactants or disinfectants like chlorine. And your water treatment process and your wastewater treatment processes should be able to inactivate the virus. The virus can survive for a few days in untreated wastewater and sewage, and therefore it is a concern. However, as I'll discuss later, the risks are very low. We do know that the virus is shed in human fecal material, in human feces, by people who are symptomatic, asymptomatic, even pre-symptomatic, and obviously infected COVID-19 patients. And what that means is that the virus actually goes through the stomach and through fecal matter and enters into sewage or wastewater. However, there's really no evidence of the infectious virus surviving in wastewater or sewage. So even though we can detect the virus, or at least the signals of the virus, there's really no evidence that it is infective. And to date, nobody has found or nobody has isolated infectious viral particles from wastewater or sewage. And to date, there is no evidence of what we call fecal oral transmission. So most of the diseases that we think about when it comes to exposure to sewage or wastewater infect people through a fecal oral pathway. And apparently for SARS-CoV-2, we still have zero evidence that this happens this way. So the bottom line is that you are safe to drink your water. You will not get infected with SARS-CoV-2 with drinking water if that water has been properly treated and disinfected with chlorine, for example. In terms of natural waters, a lot of people are trying to figure out if the virus is present in all kinds of natural waters. And so far, the prevailing knowledge is that it may be there for a few days. We can detect the RNA of the virus, but for the most part, the risks of, again, being infected are very low, unless, of course, that you have water that is directly being impacted by sewage. What about toilets and aerosolization? In other words, when you flush your toilet, do you get the spray or bioaerosols coming out of the toilet because of the flushing action? And the answer there is the risk is minimal. The number of droplets and bioaerosols that you will get from flushing your toilet, that number is actually quite small. And if you multiply that by the probability that the viruses are going to be in that aerosol, it becomes negligible. Having said that, if you have an active COVID-19 patient in your household, the guidance is that if possible, they should use a separate toilet, and there should be plenty of sanitation in terms of sanitizing surfaces. And also, if you can have a cover on your toilet before you flush, that will be good. The bottom line is that with proper hygiene, hand washing, and cleaning of surfaces, SARS-CoV-2 should be rapidly disinfected. Now, the idea that the virus is shed in human fecal material then raises the point of monitoring for the virus in wastewater. So this is called wastewater-based epidemiology. Next slide, please. Okay. So yeah, you can stay in presentation mode. Okay. So this wastewater-based epidemiology or surveillance is the idea that what you actually have in wastewater is a pooled sample. So instead of sampling each individual, you're actually sampling a whole community. If they are contributing water to a sanitary sewer or to a series of community level type of wastewater. So this is what we've been doing since April in North Carolina and also in other cities with partners in Houston, in Washington, D.C., and in California. The idea that even infected people, even pre-symptomatic, symptomatic people, and even asymptomatic people, those who don't basically show any symptoms but have the virus in them, actually contribute to the wastewater. So if you're sampling a facility that takes in the wastewater from, let's say, 100,000 people, effectively with one sample, you're sampling 100,000 people. And if you do that, and you can also sample, for example, hospitals or prisons or nursing homes or other hotspots, if you do that, you are actually saving a lot of time and money because you are saying something about the community level infection. So there have been several studies on this, and one study basically said that wastewater-based surveillance would basically save a lot of money, and the reagents themselves would cost 0.014% of testing those same number of individuals. In terms of testing, I have to note that the testing is right now mostly detecting the RNA of the virus. And again, this doesn't mean anything about the infection of the virus, just the presence of the RNA. And there are two methods that are being applied right now all around the world. One of them is reverse transcription followed by quantitative PCR. And the other one is digital droplet PCR. So my group has been doing digital droplet PCR, and we have been collecting samples since April until today. So the advantages are, next slide, please. If you are going to do this, that you could actually have different ways of using the data. For example, you can track trends. You can detect if in a community you are actually getting better in terms of the infection, or you're being hit with the second wave. So a lot of papers, there's probably maybe 10 to 12 papers that are published right now, say that you can actually detect the SARS-CoV-2 RNA in wastewater before you actually have the very first cases in patients. So we can track the trends, and we can actually predict what's going to happen in terms of if the community infection rate is going up or going down. We can also estimate the infection prevalence in the community and compare that with clinical estimates. This is a little bit tricky. There's a lot of data and a lot of assumptions that go into this, but a lot of people are working on how to actually do this estimation using models. We can look at viral evolution. In other words, what's happening to the viral genome? Do we have different strains in different geographic areas? And that will tell us how the virus has evolved over time. And there's evidence right now that actually it has evolved. There seems to be no changes in terms of how severe the virus can affect you, but it has changed. And then finally, you can do this wastewater-based surveillance to identify hotspots or areas that are have a higher prevalence, higher infection rate, and therefore you can target public health interventions in those areas. So we're continuing this work, and we are actually helping out. I'm helping out a group at the UP Diliman start up their research in terms of looking at water bodies, steros in Manila, and also some water samples in Davao and wastewater influent to do the same things in the Philippines. Thank you, and I'll be happy to answer questions later.
Thank you, Francis. This is one of those examples of the two bridges that we've been showing you early on, where in this case, I guess it's coming from North Carolina all the way down to the Pasig or Esteros in Manila. Clearly, we'd be very interested, Francis, on your progress. And I know you've been working with Carol, a fellow Araula of UP Marine Science. So clearly, if we can, well, Ayala as Manila Water, hopefully we can, and Maynilad also can be part of the whole project, I guess. But this is one of those exact synergy that we've been looking for, that's only enabled by having such a conversation between our PAASE members in the US and our local members here. So now we'll move forward in terms of our presentation. Next on deck is Chris Monterola, and he will be talking about his side of data and more on the impact of COVID-19 and what he thinks we should be doing. Take it away, Chris.
Thank you so much, Al. Good morning, good evening to everyone. Good day. I will be sharing two perspectives, hopefully to stimulate our discussion, our forum for today. Perspective one, we all know that the three main components that has been compromised because of COVID-19 is health, economy, and education. I would like to believe that this crisis, we just don't want to solve this problem, but it is possible for us to strive to create a better normal. And I'm a scientist in a business school, and we have the engagement that I have is essentially on looking. Mon, can I request that the other phones are muted? Okay. The contribution that we have is on essentially looking at data science in AI, in looking at what can happen beyond the current pandemic. So we have seen some of the reason why I'm very optimistic that there could be a better normal, that we can strive for a better normal as a result of this pandemic, is because we have seen that innovations have resulted to an acceleration in medical breakthroughs. So as Anna shared, a vaccine development that typically will take 10 to 18 years probably can now be done within 12 to 15 months. We also have seen innovation on mental health, mental depression problem, AI-enabled technology that has been unprecedentedly improved, okay, during this pandemic. Another reason why I can be very optimistic is that we have observed, and I think during this 1 month long PAASE meeting, we will hear a lot about this one, is the enhanced government agency, government academic industry collaborations. In the last 3 to 4 months, I have seen successes that I have not seen in the last 10 years, at least here in the Philippine setting. I guess some of our members, especially Al, in the next presentation, will mention some of these engagements. And third, we've been pushing for digital transformation of the different companies in the country. And there has been resistance, okay? A good portion of the local-based companies, they are not in the Industry 4.0 stage, but Industry 2.0. But now, because of the current pandemic, there is a need for them to survive, to do digital transformation. And I will be happy to expound on this during the presentation on how we help these different companies to do digital transformation during this time of the pandemic. Lastly, next slide, Mon, please. Okay, lastly, this is the second perspective that I would like to share. I believe that we must approach the crisis due to COVID-19 with an entrepreneurial lens. Okay, the main reason why we've been seeing a lot of transmission currently is that people are not used to wearing masks and PPEs. These were designed for doctors. And in general, this is too uncomfortable, especially in the Philippine setting where it is hot and humid. But more importantly, in the hierarchy of needs, PPEs and masks are expensive. Okay, in the hierarchy of needs, you can look at food, you look at shelter, even entertainment. But for classes D and E in the Philippine economy, in the Philippine setting, buying masks is too expensive. And another thing that I believe are relatively expensive too is detergents. Okay, you need to wash your clothes once you go outside, and I think that is not the priority of our countrymen who are in the classes D and E. So we need some level of innovation for masks and PPEs. And second is innovation on tracing. Current procedure in the Philippine setting is both inefficient and inconvenient. If you go to Starbucks, you need to write your name, address, and telephone number. And the veracity of that information, it is not digitized, is of course compromised. And worse, one of the things that I realized there is that you will be sharing the same pen, okay, in going from one person to another person. Maybe this is the time where we can speed up on the idea of a national ID that is RF enabled. And that is one reason why I think the anti-terrorism bill is probably not a good timing at this point in time. And lastly, I would like to emphasize that this is not just a problem on health. Okay, this is also, of course, a problem on the economy. Maybe there is a possibility, that's why I would like to entice everyone to imagine this as, of course, anchored on science, but look at it with an entrepreneurial perspective. There is a lot of unspent money from classes A, B, from the rich people in this country that maybe we can use to stimulate the economy. Unspent money or excess money simply because right now they are buying just essentials. And as a result, there are money actually that is not spent. Most of the buying of of the Philippines has been shown to be impulse buying, and now because of the current pandemic, this is not the case. The last point that I would like to emphasize is that at this point, I think mass testing is statistically not very useful now. The idea of testing 1 to 2% of the population, given the current extent of the pandemic, is probably statistically not useful in containing the pandemic. Our slim window of opportunity is over unless we can create an efficient and non-intrusive or non-invasive tracing procedure. And I will be happy to expound on the mathematics of this. I think our best chance is wasted because of three main blunders. One, we did not close our borders fast enough. Second, we allow those in the Metro Manila residents, the epicenter of the epidemic, to go to their home provinces when the epidemic is happening. And third, we have not done mass testing fast enough. I'm not sure if we have done all those three, we will be in a better situation right now because of the complexity of the Philippines landscape and the complexity of the problem. But right now, I think 1 to 2 million testing will not be sufficient to cover and actually prevent the virus from happening. Thank you. Thank you, Al.
Okay, thank you very much, Chris. I agree wholeheartedly with your recommendation. Actually, it hits me and that last point there. But we'll talk about that in a separate discussion. But clearly, to me, this is a moving target that we're dealing with, and I agree that we cannot rest until we have addressed all those issues from PPEs to diagnostic to eventually vaccination. So my whole point when Giselle asked me to do this at the beginning of our meeting was really just to describe what it's like for us, scientists stuck in our homes, quarantined, and can't go anywhere. It's been 4 months, I haven't stepped outside. What is it that we can do? And as you probably can see, we can do a lot. And so when I reflected back early on, as early as March, when the news first came out that our local kit, this is Raul Destura's kit with UP NIH being assisted by PCHRD of DOST to develop it, was just about to start their validation studies. That's when I knew, oh boy, we need a stopgap measure in terms of being able to have early testing capability. It so happened that I was part of the ASEAN Diagnostic Hub, so I gave a call to our friends in Singapore at A-Star with Dr. Sidney Yee to lend us some of their testing kits that they already rolled out as early as February 1st. And we actually had earlier conversations with Ayala Health to help us facilitate the transfer at that time. On successfully to be received as early as April, and from the initial 400 kits that they were talking about, I think Temasek Holdings, which is their national funding group, end up donating 600,000 of these kits in the early phase of the pandemic. So clearly, see what one phone call can lead to discussions with everybody else and NIH and DOST, just being aware of what is available for us, indeed accelerated that. Another example of a government industry academe partnership is on the gene expert. We found out in our PAASE website exchanges. This is the value of the communications, guys. In as much as I know you find the emails to be overwhelming, if you don't like it, just cancel it. But there are bits and nuggets of information in our exchanges that precipitated to us knowing that there are gene expert machines, 60 of them, 66 of them available in country for TB testing, and that can be easily converted into testing COVID-19 if we can get the Cepheid cartridges from California that was being just rolled out as early as April. And we work together with PBSP. This is Philippine Business for Social Progress, because they were the ones who were able to have access to the gene expert machines as well as funding to procure the cartridges. And with the help of Secretary Pernia of NEDA, contacting our DFA as well as our PBSP contacting the US Embassy for assistance and eventually getting to the Philippine Embassy in San Francisco. The whole process took a month, but in the end, we were able to order 180. And I think as of this date, Giselle can elaborate, there are 22 gene expert centers that are running, being able to conduct testing. Again, a demonstration of what government, industry, and academe can facilitate. I know Sean of UPLB was one of the early proponents into getting gene expert testing capability in Los Baños. And I heard 2 weeks ago that they opened their COVID-19 testing capability. In terms of medical device development and ventilator discussions, we had this as early as well as in March. And we were able to discuss with DOST and industry like IMI, Ayala, and Unilab Foundation about possibly being able to not only develop, but also manufacture and test. Also with PASU, the Association of State Universities, on PPE donations in the regions. This was also facilitated by Ronald and in Iloilo with San Agustin. So I just want you to open up our contacts, our Rolodex, and our connections to explore possible that connections develop in various areas, from detection to cure to even monitoring. And that's what we've been doing in the last 4 months that we are locked down. Next slide, please. So I'm just going to briefly come up with my wish list before I end the session. But moving forward, I think most of our friends are anxious to be able to go back to normal, but unfortunately, we won't get there anytime soon unless, as our earlier presenters have said, we need to have the vaccine, but even that has some questions, as Anne intimated. It's not going to be a silver bullet right away. Environmental monitoring will be critical, and saliva detection is very near and dear to my heart, and I wanted to support that, Windell. And after this meeting, we'll talk about how do we move forward with what we have and who can we contact. I've had some intimate discussions with a Japanese group, pharmaceutical, that has been implementing it in Japan. So clearly that's another area that we can explore. Definitely cures and local production, localization of PPEs, as Chris intimated, definitely is something that we should be doing. I've seen Banawe hand-woven face mask, and I've been wanting to get my hands on it so I can use it already. I bet you they'll be more comfortable coming from the north. And also definitely on the vaccine procurement and distribution and local production is also something that would need tremendous amount of capital, and this is where your AB cash in the bank or undeployed resources can be tapped, especially if we can demonstrate capability. And that's what I'm here for. I'm hoping that as a group and individually, we can demonstrate our capacity to be able to help in this fight. And in doing so, we can be beacons and shining example of what can be done. And eventually people will see and hopefully believe and contribute, open up their wallets in doing so. And as an initial uh, putting your money where your mouth is always a nice demonstration of commitment. And that's a very typical language we use in the venture capital community. And I am willing to put my money where my mouth is, as Chris has already asked us. And let's do that if we cannot do the other parts. But I'm sure as PAASE, with our collective mind power, if you want to call it in science and technology, we can do a lot. Imagine there's only 5 of us in the last less than an hour describing to you what we have, and it's tremendous already. I hope that every one of us are feeling empowered and wanting to do something to be able to win this fight. And I think with that, I'm leaving another 10 minutes available for some of our viewers as well as co-members. If they have any burning questions that they wanted to ask, I'm willing to entertain a few before Giselle gets moving to introducing our government representative before, Secretary Pernia. And I hope that you found the first hour of our 40th anniversary meeting to be productive and informative. Thank you very much.
Okay, thanks, Al. So we have 2 questions that you could address. One is from Des Hautea. What's the status of Cepheid Expert Express SARS-CoV-2? Could you give us more details? Second question is from Rodel Subade. What is the status of the kit developed by Dr. Raul Destura? Just those 2 quick questions.
That's very quick to answer. So far, Des, I think we do have in the DOH site 22 Cepheid centers that are been certified for testing. The first shipments of the Cepheid cartridges has been distributed to those centers. And in terms of the total volume, the order donated by Philippine Business for Social Progress is about 180,000 cartridges. I'm not sure yet if they've actually used up all 180,000, but based on the reports on the center outputs, I don't think they've gotten to even 100,000 of those. So I would suspect that we're still have some stock available, but with the ramp up in testing, I will follow up where we are in terms of the Cepheid delivery from US or Switzerland. So I hope. But there are 22 centers. Remember when we started this, Chris Monterola of AIM did the initial mapping for the centers to be enabled. And clearly that's when we found out that UPLB doesn't have a testing center close by. And that's what we lobbied to DOH to enable that. And that's been done. So I think I will follow up with PBSP in terms of the deliveries on the cartridges, but I'm happy to say that I think as far away as Zamboanga and Palawan are already testing. In terms of Raul Destura's team in UP, I've been informed as of today or yesterday that it is available and validated and ready to be utilized. USTU SecGeb and appeal to the scientific community to at least begin using whatever they have, if they have it available in their centers, as well as supporting it in requesting it in the various centers that they are going to. I know UP Manila, NIH, Cynthia Saloma, Eva Cochonco, and of course Chancellor Menchit Padilla are all rearing to get back to collecting the data as the tests move forward to increase our confidence in the utilization of our local. And I'm all for using our local kit, guys, because clearly this is not only the most cost-effective way, but also symbolizes our scientific effort in fighting this disease. And congratulations again to Raul and the local COVID-19 kit team for getting this up and running again and very quickly. Thank you.
Okay, thanks, Al. There's a question for Francis. It's posted. Francis, would you like to reply to the question of Dr. Ireneo Lit?
Right. So the question is, there are a lot of households in the urban poor and other human settlements that don't have proper toilets. And so fecal material may still be improperly disposed. So are there any studies on how long this coronavirus remains potent in feces? And I think the questioner, Ireneo Lit, is asking because he's an entomologist and there is no evidence of SARS-CoV-2 being transmitted by filth flies, house flies, and blowflies, but there's also no evidence that the insects, might not. So let's just be clear about what we know in terms of SARS-CoV-2. So we know it's shed in feces because we can detect the RNA of the virus. To date, we still don't have definitive evidence. There has really been no peer-reviewed study that showed that they have isolated the infectious virus. So what we're detecting is the RNA that tells us that the virus was there, but that RNA, whether or not that's from an infectious virus particle or whether that is actually just, degraded virus remnants, it's just the RNA that's persisting in the feces. We don't know. So nobody has yet isolated infectious virus from feces. So that's the first thing. So that pretty much tells us that even though people are trying, so I know of some researchers, this is what they do. What we know about the fate of SARS-CoV-2 in water are based on experiments where we actually put the virus in water and then we see how long they stay there. And that's where we say it's there for a few days. It will survive depending on temperature, depending on a lot of different factors. But we also know that they are easily disinfected. So soap will also basically destroy this envelope virus. So there is no evidence that that we have infectious virus in feces and wastewater, and therefore the other pathways like insects, for example, there's still a lot of research that needs to be done, but there doesn't seem to be any evidence of that at the moment. A lot of this is fast changing. The very first papers came out in March, and in a few months we're learning a lot, but we still don't know a lot. So thanks for the question.
Okay, thank you, Francis. Thanks to all our panelists, Anne, Windell, Francis, and Chris, who discussed critical issues and measures to curb COVID-19. And to Al, who underscored the need for academe, government, and the private sector to collaborate in order to deal with COVID-19